Hello, my name is Bruce Applegate. I'm in the Department of Food Science at Purdue University, and I also have a courtesy appointment in the Department of Biological Sciences. E. coli gets a bad name. We have to have E. coli produces micronutrients for our bodies. We naturally have E. coli in our colon, okay? This E. coli is E. coli 0157H7, which makes it a pathogen, right? So a lot of times we drop that. It tends to get dropped in the news, the 0157H7, or the toxin-producing E. coli, right? And it's important for us to remember the E. coli that we're trying to detect is a bad E. coli. Currently, the detection of foodborne pathogens, especially in ground beef samples, are dictated by the fact that we have a zero tolerance. Those organisms are classified as adulterants. There cannot be one of those organisms in the food sample, which means we constantly screen samples that are 325 grams, and we have to be able to find one bad E. coli bacteria in that 325 grams. And the problem with that is we do not have a technique that can do that. So what we do is what we call an enrichment, a selective enrichment. So we take that sample and we put a bunch of broth in there, liquid media that bacteria can grow very well in, but it also selects for the bad E. coli. So we can take that one bad E. coli and turn it from one cell into millions, hundreds of millions, millions and hundreds of millions of cells, so we know that we'll find it if it's present. And that takes at least sometimes 6 to 24 hours, and usually the procedure is done overnight. You grow the cells overnight to make sure there's enough there so you do not miss that one cell. Now, the way we've modified this virus so that it will be able to use it to cause the organism we're trying to detect to make light is it's kind of like the firefly that you see out in your backyard, except this is from an organism from the ocean, which is a shrimp that lives in the deep sea, and it actually glows in the dark. And the way it does it is it has an enzyme that it produces, and then it has like a substrate or a chemical for that enzyme. So when the two are present, you get light. So our phage causes the bacteria to produce that enzyme, and then we simply add the chemical to a sample, from the meat that we're trying to detect it in and look for it to make light. One of the advantages to using the bacteria phage or the virus that we've genetically modified to infect these bacteria is the fact that it can be added during the enrichment process. Now, most samples that are actually examined from a lot of food companies are done off-site. They are checked for the pathogen they're concerned with off-site. So usually it involves shipping, overnight shipping, which means now you have this overnight time frame where the samples are traveling to the lab to be detected. So basically, the research that we just completed showed that we can actually take advantage of that time using different packaging for the samples, add our enrichment broth, add our virus that's going to detect them, and then be able to ship it so that when it shows up the next morning at the lab to look at whether or not it has the pathogen or not, you simply have to open up the sample, add this reagent, and you'll know in a matter of minutes whether it's a positive or not.